Good morning. Alrighty, so today we are going to kind of talk a little bit about cell division. We're going to review a little bit about mitosis and then go on into meiosis. So, more doodles. And by the way, it might help if you've got two different colors, so whether it's pencil and like a blue pen or black and blue ink or something like that, just so you can kind of track everything. You can do it without it, but sometimes it makes it a little easier to see if you've got two different colors. So just kind of a review of cell division. In order to make more cells, you've got to go through cell division. It is that it's the smaller part inside of the cell cycle. There's two different ways that cells can reproduce. The first way is through asexual reproduction, which is essentially mitosis. You are making an exact copy of your parent cell, and you end up with two cells at the end that are genetically identical to that of the parent. In living organisms, when we're talking about asexual reproduction, generally we're talking about binary fission through bacteria reproduce this way. Other ways that you can technically reproduce this way and actually make more of the organism rather than, say, repairing tissue, because that's what our skin cells do whenever you get a cut or something like that. It's going to go through this asexual reproduction or mitosis to make more skin cells to seal up that cut. But it's called budding, which is where the organism it goes through mitosis and grows a smaller version of itself, and eventually that becomes a bud, pinches off and falls down next to the original parent, and then sprouts a new organism. So yeah, it's kind of weird. So usually it's going to be like aquatic animals that would do that. Things like coral reproduce this way. It's how coral reefs can expand. Sexual reproduction is going to be where you need two organisms to reproduce, and that's when you get a fusion of two gametes or two sex cells. And the way that those are produced is through the process of meiosis, which we'll talk about later. So there's a couple of more things that I want to go into with asexual and sexual reproduction. There's pros and cons to both. With asexual reproduction, the pro is that it doesn't require second organisms. So if you live in a fairly solitary environment like perhaps the Arctic or down in the bottom of the ocean where you're not mobile very much, or there's just not a lot of life around you, sometimes asexual reproduction is the way to go because you don't have to sit and wait for another organism to fertilize your seeds or whatnot. So and it's quick. In the grand scheme of things, it's pretty quick to reproduce. A con or a drawback for asexual reproduction is that eventually, because it is so quick, you can reproduce many generations in a very short amount of time. However, space can then become scarce as you get more and more organisms. So, and then obviously, since you're making an exact copy, there is no genetic variation. So when a mutation comes in, when a virus comes in and attacks one specific gene, since every single organism is genetically the same, all the organisms would then potentially get wiped out because they're clones of one another. So that's kind of a drawback. So unless there's been a mutation that occurs, that's how some organisms can survive, which that's kind of how we've gotten MRSA. There's been a few genetic variations in that particular line of bacteria that particular staph, and it has survived the antibiotics. So because the bacteria are essentially genetic clones of one another, with the exception of the MRSA ones, which have mutated, the antibiotics work on most of the bacteria except for those ones that have been genetically varied due to the mutations. So sexual reproduction, the pro for that one is it does allow for some genetic variation. So you're able to survive through diseases and viral outbreaks and things like that because you've developed those adaptations, or you've developed different versions that allow you to survive. However, it generally takes longer to reproduce offspring that way, and there's no guarantee that fertilization will actually take place. Just because the male gamete is present next to the female gamete doesn't necessarily mean that fertilization will automatically take place. So, kind of a drawback on that one. Kind of a little bit of a review of mitosis. Now, we're also going to go into a little bit more in depth, I guess. I didn't mention this, but when your body specifically goes through mitosis, that would be the division of body cells or somatic cells. Soma means body. So like I was referring to earlier, if you get a cut or a broken bone, your body will heal itself through the process of mitosis. At the end, you end up with two diploid cells. So if you recall back to our notes, we end up with two identical cells. Diploid means that they have two copies of each trait. So you end up with two copies of your eye color gene, the one from mom and one from dad, which won't be the case in meiosis. You're only going to end up with one copy. With your diploid cells, diploid is oftentimes denoted with a 2N, and I didn't put that there. Actually, it's usually a 2N, I think. Yeah, I think it's a lowercase n. So oftentimes you'll see those abbreviated as 2N. That means a diploid cell. They are genetically identical. They look like the parent. With mitosis, the parent ceases to exist once division has occurred. With your meiosis, you're going to divide specifically your gametes or your sex cells. So this is how egg and sperm cells are made. So females, once they're born, only go through the process of mitosis. They will never again go through meiosis. They'll only go through meiosis when the egg cells are developing, when they're inside their mother's womb. Males, on the other hand, since they continually produce sperm throughout their life, will go through both mitosis and meiosis throughout their life, but females won't. At the end of meiosis, you end up with 4 haploid cells, and those are usually abbreviated with a lowercase n. So instead of with 2 cells that have both copies, one from mom and one from dad, you end up with 4 cells that have 1 copy of the gene. So you'll end up with either mom or dad's eye color. You're not going to end up with both. This ensures that the offspring don't have another duplicated number of chromosomes. If we didn't go through meiosis, our offspring would have 96 chromosomes because we've got, 92. We have 46 chromosomes. If we didn't go through mitosis, our kids would end up with 92 chromosomes. And then their kids would end up with 184 chromosomes. So it would just keep growing exponentially. So that's why we want to go through meiosis. The cells are not genetically identical. They're not going to look like the parent cell, mostly because they end up with half of the genetic information. But depending on how things have separated, you will end up with different versions and different combinations of the traits. I'll explain here in a minute. It'll make sense when we draw. So with meiosis, it's going to be very similar to mitosis, especially the second half of meiosis, but the first half of meiosis is going to be a little different because we're going to be dividing pairs of chromosomes. We're not going to be dividing the chromatids themselves yet. So in order to get 4 cells, you're going to have to go through division twice, because the first round of divisions, you're going to end up with 2 cells, and then those 2 cells are going to go through division to end up with 4 cells. Okay? So it's... sorry. It's not the next slide. So, kind of an overview with meiosis. This is where we produce our sex cells. Like I said earlier, the reason why we go through this is to make sure that the next generation doesn't have too many. So you start with a diploid cell, because you've got diploid, meaning you've got 2 copies of each trait. You're going to end up with a haploid cell. Specifically 4, but a haploid cell, meaning each cell is going to have only 1 copy of each trait. So you'll start with 46, and then your 4 different gametes are going to end up with 23. I said chromosomes here. Technically, that's incorrect. It would be 23 chromatids. And then fertilization will then restore the diploid number. Because you'll take the 23 from the sperm and 23 chromatids from the egg, and they'll recombine, so you end up with 46 chromosomes. And like I said, do note that I'm technically using chromosomes incorrectly, because you won't have chromosomes at that point. You'll have pieces of chromatids. And like I said, you end up with 4 haploid cells at the end of meiosis. So I've got a few doodles here, but I've also got the doodles on your sheet of paper also. So it'll kind of help you in both. But like I told you earlier, meiosis involves 2 different cycles of division. The first round is called meiosis 1. The second round will be called meiosis 2. Meiosis, they're both similar to mitosis, but meiosis 2 is more. I think it looks a little bit more, because you end up with a piece of chromatid instead of chromosome. So because we're going through 2 rounds of division, the individual steps inside of meiosis 1 are going to also be assigned a number as well. So you're going to have prophase 1, metaphase 1, anaphase 1, telophase 1. Then when you go into meiosis 2, you'll go through prophase 2, metaphase 2, and so on. So there are going to be some similarities and some differences here. In prophase 1, I don't have it on this PowerPoint slide, but if you look at your drawings, I've included the very, very first step where it shows you what you started with. So you would have ended up with 1 long blue piece, 1 short blue chromosome, 1 long green. I think I drew it as red. 1 red chromosome, and in this case it's green, because that's what the picture was. That's the one I could find. And then a short green. Then, through the cell cycle, specifically through the S phase, those pieces of chromatids tightened up, replicated, and became chromosomes. And so this is where prophase starts. Okay, so they've already been duplicated. Don't forget that little spot there in the middle, that's called the centromere, holding those 2 sister chromatids together, so that this side right here is identical to this side. Okay, so what happens in prophase 1 is they've already duplicated, and we're going to call these homologous chromosomes, meaning they're similar in function, but they're going to be different versions. So these larger chromosomes might code for eye color, and then these smaller ones might code for hair color. In the grand scheme of things, hair color and eye color is coded for by more than one chromosome, but to make it easier, we generally, especially at the high school level, just say, well, to make it easier and simpler, we'll just say that eye color is coded by one set of chromosomes. But in all actuality, it's not. So we've got these guys have matched up because you don't want if the bigger ones, I can't remember, I think I said eye color. These code for eye color and these code for hair color. You don't want the eye color code, matching up with the hair color, because then you won't necessarily end up with a chromosome for eye color and hair color. At the end, you might end up with 2 copies of eye color, which would be bad because then you'd have no hair color. So and that's what homologous means. Similar in function, so they're both coding for the same thing, but they're just different in what they actually are. Obviously, one is from mom, one's from dad. We'll say the red one's from mom, the blue one's from dad. So they pair up. This process is called synapsis. When you have them in a pair here, we're going to refer to this as a tetrad. So we've got the hair color tetrad and the eye color tetrad here. Okay? Nuclear envelope and nucleolus are still dissolving. The centrioles are forming. Spindle fibers are still building. So all of this is just the same as it was with mitosis. But what can happen here that's different is there's this thing called crossing over. Crossing over can occur. And I don't know how well you can see it on the little guys, but on the big guys, you see how on the picture, the green, which is the red, is kind of crossing over and laying on top of the blue here. This is sometimes how we get slightly more combinations, because what's going to happen if you look over here, and I don't know how well it's showing up on this one, but I think it showed up pretty good on the ones that I drew you guys. You can see how this side is all one color, which on your sheet would be red, but up here it's green. But then over here, and they've crossed over in 2 places here. But then over here, this is not technically an exact duplicate of the left side over here. This now has a little bit of the blue at the very top and a little bit of the blue chromatid down here, and vice versa over here. This has a little bit of green down here and a little bit of green up here. So that's just another mechanism for more genetic variation, is what crossing over is. And it happens up here in the hair color chromosomes also. So this is where crossing over can occur, when you are recombining your genes. Then we're going to move into metaphase 1, where you are going to get your chromosomes to line up at the metaphase plate. Now they're going to line up to where they're on, where you've got one pair or one part of each pair is on either side, because the division is going to occur this way, from the top to the bottom, not straight across laterally. Because then if you did that, this cell up here would end up with 2 copies of the hair color cell and no copies of the eye color, and vice versa. Down here you'd end up with 2 copies of the eye color but no copies of the hair color. And your body, it's crazy, but your body knows how to do this. It just does it automatically. And so the split will occur from top to bottom like this. So you end up with 1 copy of each trait there in each of the cells. The spindle fibers will then attach to your centromeres. So remember, the metaphase plate is the center or the equator of the cell. And you're going to go through anaphase 1, which is where the spindle fibers are going to kind of pull apart your chromosomes here, and you're going to get kind of the cytoplasm starting to pinch off here. But again, you've got one version of the eye color, one version of the hair color. So they're going to the poles. Telophase is going to occur, where your cell membrane is going to start pinching off. You're still going to get those little pectin-filled vesicles, and you're going to still get the plasmodesmata and stuff like that. And then cytokinesis is finished. Your cytoplasm is fully divided, and you've got 2 cells. You've got 1 chromosome for each trait, but remember, you've got technically 2 copies. So in this case, you've got mom, in this cell on the left, you've got mom's eye color, but you have dad's hair color. And over here, you've got mom's hair color, but dad's eye color. But you've still got two copies. So we're not really diploid anymore, but we're not really haploid either. So we've got to go through the next set of divisions, which is meiosis 2. This is, I think it looks similar to mitosis, I think, a little bit more, because you end up with a chromatid at the end. But so we get get new spindle fibers and the centrioles and everything. So again, they're going to start moving towards the metaphase plate at this point, but they're technically already fattened up or condensed, so they don't really have to do that again. Metaphase 2, they're going to line up with those metaphase plates there. The spindle fibers will attach at the centromere. They're in the center. And then we're going to move on to anaphase 2, where the centromere split apart. Now it looks similar to what you ended up with back in mitosis. I think it looks more like it. And the cytoplasm is going to start pinching in, because you're going to start getting this cell dividing and this cell dividing, so you're going to end up with those 4 cells that you want to end up with. So your chromatids are moving to opposite poles. Telophase starts, and you're going to, it didn't show it up here, but I did in your diagrams, you're going to start getting the nuclear envelope reappearing. The nucleolus and everything. And then cytokinesis occurs, and you end up with 4 haploid cells. So you end up with 1 copy of your eye color and 1 copy of your hair color. Now, if crossing over hadn't occurred, these 2 cells would be genetically identical. These 2 cells would be genetically identical. But since crossing over did happen, none of these are genetically identical. None of them, regardless of whether or not cytokinesis occurs or not, are genetically identical to the parent, because you end up with half. Each one of these cells is haploid because you have 1 copy of each. You don't have 2 copies. You only have mom's copy of eye color here. You don't have mom and dad's eye color, just mom's. And over here, you have just dad's hair color. You don't have mom and dad's. That's why it's called haploid. So again, kind of recap and refresh. Meiosis is going to ensure that we get some genetic variation within the offspring and the species. They're going to, the offspring will receive different combinations. Crossing over intensifies that genetic variation, or the different combinations, if it occurs, which it does often. And think about how small your nucleus is and how tight all those chromosomes are. It's very common for that crossing over to happen. And then that ensures that each offspring will have a diploid number of chromosomes and not more than diploid. So it makes sure that you don't have 4 versions of each chromosome or each gene. So I know that was a lot. Hopefully the pictures kind of help. I mean, look at the end result and compare it to that of the beginning, and you'll see that it's definitely half. So again, if you do have any questions, don't hesitate to ask. And that kind of wraps up meiosis. So again, questions, just holler. And have a great day.